Okay, this video is going to talk about the CRISPR Cas9 system, which is interesting for two different reasons. One, it's provided us with a really powerful genetic tool that we could use to modify eukaryotic genomes in ways that we haven't been able to do before. And two, it actually just the innate function of this system is really interesting because it's essentially analogous to an adaptive immune system within bacterial cells. Of course, it's not, it doesn't function in any way like our adaptive immune system, but it is adaptive in that it allows that bacterial cell to respond to foreign invaders based on previous experience with those foreign invaders. And so how does this work? And by foreign invader, I mean, like a virus. And so if we have a little viral particle right here that injects its DNA into our bacterial cell, now provided that, if the bacterial cell ends up dying, then this isn't going to work. But provided that the bacterial cell can actually get to this DNA before the virus ends up, killing the cell, then that viral DNA can be chopped up into short pieces called spacer pieces, and that's done by these Cas9 proteins that are encoded within the bacterial chromosome. And now these spacers, which basically are these short sequences from the viral DNA, are going to be incorporated into this CRISPR region of the bacterial DNA. And so what CRISPR stands for is clustered regulatory interspersed short palindromic repeats. And what that refers to are actually these little black segments here, which are these repeats that, repeat over and over. But the actual sort of moneymaker of this system are not the repeats themselves, although they are important, but they're actually the spacers in between the repeats, because each of the spacers represents a different part of a viral genome or a different viral genome that can then be used to identify further infection by that same virus. And so basically the first part of this process is the virus infects the bacterial cell. Bacterial cell cuts up its DNA into these short little spacers and then puts it into this CRISPR array. Okay, so then what happens if we have a second infection by the same virus? So let's take a look at that. Okay, so now we have our second infection. So we have another viral particle over here that injects its DNA. And in the meanwhile, what's been happening is that this CRISPR array has been getting translated, or not translated, but transcribed. And so we've got this CRISPR RNA array that's made. And then there's a few other components here that are also transcribed, and some of them translated as well. So one is this tracer RNA that needs to be made for this process to work. And so that also works as RNA, so it's not translated. Then Cas9 is a protein, so it's transcribed and translated. And it has regions that will bind to RNA, and it also has nuclease domains that can cleave DNA, and I'll tell you how that works in a sec. And then we also have this RNase 3, which is also a protein. And so now all of these things have to come together, and it forms this long sort of array where we've got our Cas9 bound to each of the spacers and repeat regions. And then the repeat region, which is this area in black, that hybridizes, so it base pairs with our tracer RNA. Right? So each of these Cas9 complexes includes the spacer right here. It includes a repeat and includes tracer, which is bound to the repeat. And now our RNase 3 cleaves these guys so that then we end up with a mature Cas9 protein that includes the repeat hybridized to our tracer RNA and then also the spacer. And remember that the spacer is then going to represent the viral DNA from the previous infection. And so it's going to hybridize to that viral DNA. So now if we have a second infection, then if this spacer region can recognize the DNA from that second infection, then you end up with hybridization to that DNA. And then now this Cas9 complex will actually cleave that new viral DNA that was just put into the cell. And so again, this is a way for the bacterial cell to be able to use previous infections to be able to identify viral DNAs and then degrade those DNAs through this complementary base pairing between the spacers that were generated from the previous infection and the new target DNA that was just injected. And so that's pretty cool. I think. But what's even more cool about it is that after the system was sort of first identified, some of the scientists that worked on it figured that they should be able to use some of these components to modify eukaryotic DNA. And and you should be able to do this because eukaryotes don't have anything analogous to the CRISPR-Cas9 system, so they don't have any of these proteins. And so if you insert the proteins into the eukaryotic cell, you are able to modify just the region that you're interested in. It shouldn't have an effect on any of the other eukaryotic systems. And so what would you need to be able to insert into the eukaryotic cell in order to actually use this? So as it turns out, you really don't need all that much. So you need Cas9 because that forms this complex protein thing. You actually don't need RNase 3 because you don't necessarily need to insert this entire long RNA thing. You could just insert one RNA that essentially represents the stuff that's found in this mature complex. And so really, to utilize this in a different organism, you just need to put in this Cas9 business and then this modified RNA. And so, I told you before that this RNA is actually two RNAs, but researchers can actually make one RNA that sort of does the function of both of these. And so let's take a look at that. So if we take a closer look at our sort of moneymaker in this system, we've got our Cas9 protein in blue, and then you can make this sort of RNA that both has the spacer region and has the repeat region, and also has the tracer RNA, but now it's all just one RNA, right? And so if you want to use this as a researcher to identify a particular region within the DNA or a particular sequence in the DNA, what you would do is you would make an RNA that includes the tracer and the repeat, and then this part in blue, the spacer region, you just put in whatever sequence you're interested in that you're trying to get this thing to target. And so yeah, that could be pretty much just about any sequence. And these spacers are pretty long. I mean, they're not super duper long, but they're pretty long to where you can make them very specific for just a particular sequence you're interested in. And so that's great. And so what can we do with this? So if we just did that, you've made your, you've gotten your eukaryotic cell, you've inserted your Cas9 protein, you've inserted this RNA, and it has some sort of spacer that maybe has a sequence for, I don't know, the hemoglobin gene or something like that, right? Then if you insert this into the cell, what would happen is that the RNA would associate with our Cas9, you'd recognize that gene for hemoglobin, and then this Cas9 protein would cleave at that region. And that could be really useful because once you make a cleavage there, you could potentially accumulate some kind of mutation or something like that to knock out your gene. But you could actually use modified versions of the system or multiple copies of the system in order to be a little bit more specific about the downstream effect. And so let's look at some of the ways that this system can be used in combination with like other Cas9s or with modified versions of Cas9. Okay, so the original sort of system kind of just does this. So it would do a double-stranded cut at whatever DNA you've targeted. But that, it's possible that would just be repaired with the regular DNA repair enzymes. So maybe what you want to do is actually make sure that you're cutting out like a larger region of the DNA. Well, you could do that if you insert Cas9 and then two different RNAs that target two adjacent parts of a gene. And so then if you cleave through both parts of these, then you would end up deleting this middle section. You could also put in a Cas9 protein that has a mutation in one of the nuclease domains so that you just get a single cut in the DNA, so just one of the strands is digested. And then you could use, again, a combination of these proteins that have this type of mutation so that you can cut through one strand over here and one strand next to it over here and end up with sort of sticky end cuts of your DNA. Besides this, so all these things could be super useful. You could use it to remove genes. You could use it potentially to even insert genes, although that's a little bit harder to do. But you could also modify Cas9 in other ways that could make it useful for other types of functions. So, for example, there have been some groups that have tried to fuse like a transcription factor or something like that to Cas9, and then you could use this system to actually target particular promoters for activation by using the fused transcription factor activation domain to bring in a polymerase, for example. Some groups have also tried to fuse the Cas9 to something like GFP maybe, so that you can see a particular region of the DNA. And I actually just went to a talk last week where there was a fella that was talking about trying to do this. And apparently it's actually kind of hard to get it to work, probably because the interaction between the Cas9 and the DNA is really transient. So it comes on and off a lot, so you don't really get a very stable interaction. But still, works well enough to cleave the DNA, I guess. Then potentially you can add some other types of nucleases to Cas9, although why you would need to do that, I'm not sure, but apparently this review article says that that's a thing that people do. And also you could add proteins that are going to promote recombination, and so you could do targeted recombination using this type of system. And so it's actually, really interesting from the perspective of that is sort of this adaptive type of immune character to it for bacterial cells, but then also it's been used by scientists to really kind of further our capabilities of manipulating eukaryotic genomes, which is kind of cool as well. And if you're interested in this subject more, then I could recommend some review articles for you. Thanks for watching the video.